in there.
Yep.
Nice.
Okay, back to the flush jar there. Dredge is off, right?
Thank
I
you.
mean, suction's off.
Yep.
Yeah, our pleasure. Is that it for suction?
That will be it for suction samples.
All jars are
We're
full.
all full.
Yep,
Sweet.
awful.
We are flushed with sponges now. Flushed, not flushed. We are flush with sponges. That makes more sense.
I want to switch over. It looks like the flush is still going. Can you verify it's off, Jake?
Yeah, it's off. I just turned it off.
And right. Okay.
If the idea is to do a transect, I think a good bearing would be 350. That's upslope to the top of this peak here.
That sounds
Roger.
good. And we're past the 100 meters since the last transect end.
Yes. Just
awesome.
past it, 105 meters. It will be, it looks like it's
bubble
about,
cam on one.
looks like it'll be a rocky varying slope. So you want to go 0.3
Sure.
or 0.2.
Looks like it's vertical at the moment, so
Yeah.
we're close to it. I don't.
Yeah.
Coming up. Does this begin transect, is it?
Yeah, we can call it that. Actually, by the time I get the ship moving, you'll have some ground covered, I think, vertical. Bridge nav.
This will be transect 9.
Yes, transect number 9.
Awesome.
Step 100 meters, bearing 350, speed 0.3. Thank you.
You have to come up with a Jake.
Coming up.
You
Bridge nav, do you copy?
tilt up on Argus a bit. That's good.
Bridge nav.
They all went to lunch.
No, they're blinking at me. I just can't hear them or know that they're hearing me.
Getting a little more paragorgia there, so I think we are getting into an area that's going to have more coral.
Bridge nav, do you copy?
Fingers crossed.
Hold what you got there.
Steve, we might have to sort out this audio issue.
Copy that.
We're gonna pause transect.
Yeah, we'll just say it hasn't started yet.
Do you want me to run up to the bridge?
Yeah, that'd be great.
Sure.
I'm on comms here if you want to try me to
Yeah,
troubleshoot.
I'll pop over.
You try the radio.
They usually don't have it on, but we might have to switch to that.
Nice little sculpin inside that sponge.
You copy, Bridge? Yeah. We'll do a step 100 meters bearing 350, speed 0.3. Yes, thank you.
They sounded all right. I missed that exchange.
Yeah, it sounds good.
Okay.
I'll just make sure that they're okay up there.
Okay. Am I good to call this the start of the transect?
Yeah. Start of transit. Thought we already started. We paused there.
Is this 9?
Yes.
Okay. Go ahead, Bridge. Okay, yeah, no worries. Thank you. Thanks for sorting it out.
Nice long nose skate there.
It's a boy.
Mm-hmm.
We've got some great terrain that we're traversing. We've got lots of big rocky outcroppings of this boulder-like substrate. Lots of cool places for corals and sponges to attach.
Seeing some little red swiftia starting to appear on these rocks. I think that's a black-gill rockfish, but I'm sure Tom will correct me if it's not.
Oh, I missed his science chat. He said there was a sculpin we should have zoomed on, but I didn't see it.
Yeah, I didn't see it till we were already in motion on the transect again. He says it is a blackgill.
You'll see the white stick with the almost looks like it has a ball on it to the left. That's another Paragorgia species of coral. And then you'll also see this white frilly arms, these white frilly arms attached to the rock, and that's actually a sea cucumber, a stationary sea cucumber, and those frilly pieces are its feeding parts. It will filter feed the water and clean whatever gets stuck to its feeding appendages. We're also seeing a lot of dead sponge, which other sponges will attach to and grow on.
So as we're moving up to the top of this other feature, we are moving into the area where the habitat suitability model predicted many more coral species. So far, that seems to be panning out.
Cody Point Redemption. So if you're just joining us, we are in the Channel Islands National Marine Sanctuary, and we are south of, just between Santa Cruz Island and Anacapa, in an area that's a bit deeper southeast than the Footprint Reef, if you're familiar at all with that area and that marine reserve. We're outside the reserve right now, and we're doing some quantitative transects here to get a better idea and quantify the various corals and sponges and habitat types that we're coming across. The Footprint Reef is an area that has been surveyed very well, but with new mapping data and some habitat suitability model predictions, we noticed that this area that, as far as we know, have had no surveys on it. So this is the first time a lot of the scientists are seeing this part of the seafloor, and we're hoping once we get to about probably shallower than 400 meters, we'll be seeing a lot more deep sea coral and sponge species at higher densities. But we've been seeing a lot of sponge species up to this point, sponge gardens almost. I think we've been calling them for the last several hours.
The whole first part of this dive has all been these really elaborate sponge gardens that started at about 700 meters and have continued all the way up. Oh, we've got a nice bigger Paragorgia specimen here.
Oh, wonderful. Promising.
Mm-hmm. Got all its feeding polyps out, so it's giving it that fuzzy appearance. And then more of these Anthoptyllum sea pens. We sampled one of these earlier, so that when it gets to the surface and then goes back to Gary Williams at Cal Academy, he'll be able to take a look and find out whether or not it is potentially a rock pen and potentially a new species.
You want to give us your comms spiel now, Steve? Sorry, didn't mean to cut you off there earlier.
No worries. I was just reminding people if they're going to adjust their headset or take it off and put it down on the table to please mute themselves. And yeah, beware if you're chewing gum or having a snack that we're hearing all that too. Thanks.
I'm going to pop off for a second for the dive brief for the upcoming dive. I'll join in about a half hour.
Hey.
You'll be in good hands with Meredith. Thanks.
Started in that very vertical habitat, and it still seems pretty steep, but we're definitely into less rock wall and more boulders.
Mm-hmm.
For anyone watching along, we're currently on a quantitative transect. This is a method we've been using throughout the season here on Nautilus in multiple sanctuaries, where the pilots are flying at a steady speed at a pretty constant altitude over the seafloor. And what this is going to allow us to do is take this video back to Southwest Fisheries Science Center, where analysts will watch the video and count all of the invertebrates and fish that are over 10 centimeters. And then because we've used the same methods coast-wide, we're able to compare the different locations where we're diving and get a better kind of quantitative picture about the habitat types that the corals and sponges and their associates are inhabiting. Getting back into more sponges again. A really cool rock formation there.
Yeah,
Really complex.
I cheated a little on the transect there to light this up. It was showing up in our sonar there. Couldn't resist.
I was wondering what you're breaking off for.
It'll just be the new transect heading.
Yeah, can do. Is that it? That's okay?
It'll be a long contour
Yeah.
if you want to change, rather than upslope.
I'm just following the little formation here. The hiking. I think I'll be able to come back around on the.
Okay.
It's kind of the outcrop, you know, so the current's blowing up over it. So you get more things to look at.
Nice big rhabdoid with the spiral sponges. Ooh, and we are definitely seeing more coral. The little red fluff is becoming more and more abundant.
You can see the zone there where the current comes up over the ridge. It's all nothing to the right and a whole bunch of stuff to the left.
Do you want me to redirect here? Because otherwise Argus is going to head up to that.
Yeah, let's step 20 meters that way.
Bridge nav.
And I can follow this up. Or 30 meters, whatever.
Step 30 meters, bearing 270. Sorry, 315.
Nice pile of urchins there. Looks like they're feeding on some kelp
Thank
ball.
you.
I always find it exciting to like watch what's coming in the distance and you can't quite see what it is. And it's like, what is it? What is it?
Hey, Rennie, could you drop a target at where that kelp was? Like, I guess just right here, just
Right
in
here?
case. Yeah. And that's interesting that there's a piece of kelp with a whole bunch of urchins on it.
There was.
Thank you.
Yep.
I'm trying to see if this white object that we're coming up to is coral or sponge. I think it's
There's a ridge there.
coral-like.
Yep.
My spiral sponge that's growing in basically a U shape right there.
I'm running out of leash there.
Yeah, you're
Bridge nav. Hold this move and then let's step 30 meters, 255.
Let's pause the transect here for we move the ship over a little bit.
Yep. I'll make a note in our notes. Tom just commented, Herc can't pull the ship. I thought it had power.
It can, but it makes the navigator and the Argus pilot aggravated.
Yeah, I spin around to nothingness.
I can't imagine that the video likes it much either. Because I can't imagine the video stays still at that point.
No, it becomes a little, it's one body system at that point. It doesn't seem to be helping me. So you have to come to the south.
You should be going this way, a little bit up slope, right? Because we're going to follow this.
Well, I'm basically looking west there, so you need to come to the south a little.
Okay. We're going to come into some flatter terrain up here.
I know, but I can't proceed on this heading right now.
Okay. Bridge nav.
If that's correct.
Yep, hold position there, and let's step 20 meters south. Thank you. Should be able to drive forward.
Still pulling on it really hard.
It'll ease up if you go forward, I think.
Yeah, roger. Okay, resuming transect.
All right. Thank you. We've got a nice eelpout or a hagfish. It's a hagfish in that curled up body position. Oh, and we've got a little Cumorella of black coral just going off the bottom left of the screen.
Lizzie's joining you guys again. Hello.
Tom said that was a small Christmas tree coral we just went over.
Yeah, I saw a fluffy little gray guy go down towards the left as I popped on.
Got this nice kind of white knobby barrel sponge here. I'm not sure, this might be one of the ones that Abby calls an IKEA lamp. These nice pointed knobs that come off of it.
Bridge nav. Five 0 meters, bearing 295.
Got another nice big paragorgia coming up. On yesterday's dive, we were seeing two different color morphs, a really dark pink and then these more white individuals as well. Those are likely to be two different species that are overlapping here. Today, so far, all we're seeing is this white Paragorgia, which is at this step probably Paragorgia stephencairnsii. It is a genus that is still undergoing a great deal of taxonomic revision right now. So we've been sampling some of it to help get a better picture of what the local species distribution is. And Paragorgia is also a group that we're interested in doing some population connectivity studies on.
Some really interesting rock formations here.
Yeah.
Yeah, I
really
was just looking at that.
I'm like thinking
Almost like
of
a
words to
pavement.
describe it. I'm like, it looks like a keyboard, I guess, because that's when it's like broken up in squares.
Very geometric.
They come up to 2 meters to get over that sponge.
Yeah. It'd be quite tall.
I'm assuming, Meredith, you saw the floofy Paragorgia from Olympic Coast?
Floofy Paragorgia. You mean the gigantic Paragorgia fans that had all their feet pumped out?
Stop him up for a minute.
Yeah.
Great stuff.
Those were really impressive. They were really nice to see.
Hold position.
I don't think we've ever seen a specimen that look quite like that here. I wonder if they're just had extra long polyps, or it was just because they were so large in their size, their polyps are that much bigger and impressive.
Probably a little of both, and they probably had them just really fully extended to get out in the water column. Oh, we have an octopus just going off the bottom of the screen. It's far one flapjack octopus that I know of this dive. Must have saw another one while I was sleeping.
We're currently around 413 meters deep. I think we'll still be climbing a bit. The shallowest point should be somewhere around 200 meters at the top of this feature.
Yeah, we made redirect to go back up north.
We're slowly still climbing, but
yeah,
should go vertical here in 10 meters again. It's definitely more vertical to the north, but I think that's where the current's kind of
interesting to see these really sandy channels kind of between these deeper rocky features.
All right, so you still want to head in this direction?
Yeah, just for another 10 or 20 meters, I think.
Bridge nav. Two 30 meters, bearing 300. Thank you.
So I did upload or add a shot of the backscatter to the dive plan. And if you kind of look at what the models are predicting and where the highest diversity of coral species land, there's definitely like a a stronger return, indicating harder habitat right in those areas. But there's also like a vein of it just to the west that runs parallel.
I can turn right here, Rennie. Follow this one up.
Roger. I'll let him get a little closer and then I'll follow you up.
Roger.
Would it be possible to get the nav back on high pack?
Yep, there you go.
Thank you.
Looks like it's
As
going
much
to
as
be
I love looking at Anacapa.
a blank shelf here for about 10 meters or so, and then hit some more vertical.
It'll be variety for everyone who's analyzing the video. Can't give them nice hard bottom all the time.
No. Have to go downhill at some point if we do that.
Yeah, I knew this was coming.
Got some nice sea pans here though.
But see the way the wind's blowing there?
Yeah.
So that kind of all but followed the ridge line there for a while. It was boring on the down breeze.
Bridge now.
Now I'm paying for it.
Let's go 100 meters north.
We got all
Thank
these
you.
sea pens laid over.
Yeah, it's quite ripping through here.
Are these the grandiflorum?
Yeah, these are grandiflorum. This is the first dive I've ever seen them in quite this high numbers. Like you see them a lot on different dives, but I haven't seen big fields of them like this. I've known they're around. I do some work with the trawl survey, and they occasionally will just get a net that's nothing but these guys.
Wow.
They're hanging out on the ridge line there, enjoying the breeze.
Probably optimum zone for feeding. Letting the current do all the work, bringing the food to them.
Starboard box seems to be happy now. Closed. Don't know why it was creeping open.
Something to do with Jake's button, I think.
Yeah.
Maybe they did a reboot at some point during the dive, I'm told. Kind of aiming for a V here. Hopefully it will be doesn't look too steep though.
Can I get a rough flat long, maybe typed into the science chat, just to see, kind of want to plot a point on the map to see where we are relative to the backscatter that I have up.
It's also up on the high pack under Hercules lat long, but I don't know if it's garbled
my
for
goodness.
you.
Oh, perfect. No. I can work with that.
Yeah.
Thank you.
More cobbles mixed in with the sand now.
Yeah, it's changing a lot. We've had a really diverse array of habitats just within this transect.
Yeah, we're starting to get a little return on the sonar.
Yeah, sorry, didn't get to put the backscatter up between watch last night and watch this morning.
No, you're totally fine. Oh, cool. All right. Yeah, we're approaching the ridge line that looks like a pretty hard return. That's just west of the 3 points 8234 that I was interested in. So this is perfect.
Perfect, I'll have to do.
And we'll be able to tell Matt at the end of this dive that he has Pody Point redemption.
Yes.
Hmm.
Yeah, those sponge gardens were pretty spectacular. I'm glad we're filled up to the gills with sponges samples.
Yep. So one of the efforts that's supporting the analysis on this cruise is the West Coast Deep Sea Coral Initiative, which both Lizzie and I are a part of. And at our opening workshop, it's 2 years ago now, getting a better handle on the distribution and taxonomy of sponges up and down the West Coast came out as a pretty big priority. And so it's great to be able to do these collections. And we'll really be able to build on what we know about sponges and hopefully start giving them names just beyond sponge on ID in our various databases. And that in turn will help us with the habitat models. And they're just cool and pretty to look at too.
Is that a tiny Paragorgia?
Yep. Yeah, and we'll get into some areas we found on some dives where you'll get just a bunch of Paragorgia that are all like 3 inches high, like they've all come from the same recruitment event or something.
Hmm.
That shouldn't happen on channel 2 anymore. Our side tone.
That was my radio was set to video, or one of the channels was set to video, so I switched it back.
Nice spiny king crab there.
I don't know if that that tone thing was done to the video channel as well.
No, I think just
1 and 2.
I'm not sure if we even did 1. I think we did the
I think it was just 2.
Oh, okay.
noisy one. You have to ask Paul if he did that.
Yeah,
What tone are you talking about?
the radio.
Side tone,
Yeah,
so they don't squawk when you walk in the hangar.
it's like a inaudible.
Oh.
He was supposed to do a write-up on that, and I don't think he did.
check in with him.
It's an odd rock.
Yeah.
Oh yeah, just kind of hanging out.
Somebody rolled that down the hill.
Hopefully with that sponge, it doesn't look like it's moved in a while.
Hmm.
Two more header polypus mushroom corals here.
You'll have to stop him for a minute or slow him down. I'm
Roger.
having trouble keeping out in front
Bridge
there.
now. Hold position. And I think we've just about covered it. Let me just do a quick measurement because we deviated, so I think we covered the ground. 60, 75, 13 and a half, and 18. Yeah, should be good. We can call this the end of transect.
All right. So if we could pause actually and take a Niskin bottle, that would be great. Because then I'll have one paired with that transect.
Sure.
What? Oh, that's why it's open. Oh, for
The rocker.
jeez,
The rocker was set to. That was probably because of the topside restart.
It was.
Yeah, that makes sense now.
Oh, that explains it.
Oh, it wasn't you, Jake. It was me. Don't.
At least it can be explained.
Yeah.
It's much better than the alternative.
Not gremlins in the machine this time.
Yeah.
Looks like we're on Niskin number
Number 4 is open.
4.
Yep.
Steve? Countdown?
3, 2, 1, fire.
Okay.
Nice.
And the sample number?
82082.
Roger. Oops, I just rewrote something.
Thank you.
I'll go all the way to the left. Seems like it's sticking in more than it usually does.
It looks like it's in its normal spot.
Yeah, Roger. Still headed north, are we?
Yeah, do we want to keep progress there, Science, or any other tasks here while we're paused?
We'll just keep an eye out as we're proceeding, and I'll try to get of you enough warning that if we see something to sample, we can pause as we go along. But I'm not seeing a lot of coral here.
Roger.
So, and we're full up on sponges, so I'll just keep an eye out as we proceed along.
$100 meters north. Thank you.
We'll also keep an eye out for more kelp and urchins.
Copy. Just waiting for the boat, so I'm wagging my tail around, looking, having a look.
That's a nice paragorgia.
Yep.
Bubblegum coral. Is it
People also
bubblegum
call
coral
those.
because? Oh, sorry. Go ahead.
Oh, some people also call those, I think, candy cane coral.
Mm.
Or peppermint coral, that's what it is. Just because it's white with the pink dots.
Got it. I was wondering, might be a dumb question. Is the reason they're called bubblegum corals because some of their morphologies or some of the species are bright pink? Or is it because they look like sticks with like wads of chewed gum stuck to them because they're kind of bulbous?
You know, I'm not
both?
actually sure. My guess is that it's both, since they look kind of like chewed bubble gum, but I don't actually know.
Because some of them are pretty pink. They range from red to pink, and then these are white without the polyps extruded. So I was just wondering. Seeing an urchin, also keeping your eyes out for some kelp to see if we can do a double collection at some point.
Sorry, what do you want to collect? I didn't hear the whole thing there.
Oh, no, I was just saying I saw an urchin, so I'm hoping at some point we might see some kelp that we can potentially, well, actually, do we need, Taylor Ann, are we out of slurps?
Yeah, we
We're
are
out of slurps.
Yeah, I was like interested in that one cluster, I guess just because there were actually urchins feeding on that piece of kelp, but yeah, it doesn't matter.
Gotta
yeah,
zoom
I
on the
think
octopus.
a higher priority would be urchins over the kelp.
Okay.
Yeah. So if we see a cluster.
Okay, good to know.
Yeah.
Nice.
Looks like it was thinking about running away and then was like, I'll just curl up and get more camouflaged.
They're brooding or eating?
Might be eating something. Brooding, they're usually down in a cave.
Yeah.
Yeah, he's got some tentacles in a hole. He could be hunting.
Maybe he's fishing.
Yeah.
Or she.
Yes, he or she.
Do you want me to pause here?
Sure. Pause and then I'll get back out in front.
Bridge now.
See how long we can sit here until somebody complains.
Hold position.
Come on.
More paragorgia. They're getting bigger and more
They
abundant.
are getting bigger.
It's a red one in the
it looks like
background.
we got a dark pink one in the background.
Yeah.
I'll slide over there. So I've got a row of them here right at the outcropping.
Yep.
Do you want a close-up of the red one?
Yeah, that would be possible. That would be great.
Can do.
I'm going to turn around and get you in view.
No, it's all right. You can hold what you got there.
All right.
I'll get out from underneath you. Rennie can back the ship up a little bit.
Sure. Bridge nav. One 5 meters south.
Oh, it's nice and floofy.
Yep. The polyps out. Probably enjoying the high current, bringing some food.
Okay, can try and zoom in there, Steve. I don't know if I got it stable or not.
That would be a nice specimen to print.
Right.
Mm-hmm.
Or hold sideways in that current on
The MCZ let me know that I can get loaned some of their specimens, so I'm gonna have to do some digging to see if there's any aesthetically pleasing paragorgia like this.
Might be able to get away with a pull-up zoom there now.
Nice. That's good for me. Thank you.
My pleasure.
Back north.
Yeah, let me scoot out front just a little bit there.
Yeah, I could also go 0.2 and then we'll have some time.
Maybe that's better. Kind of zigging and zagging here. And
Yeah. Exploration speed.
interesting
Bridge
things
nav.
to look at.
Let's go north 100 meters, 0.2. Thank you. Gives you some chance to wag back and forth.
Yeah, trying to find an outcrop to follow to the north.
Not seeing much in Argus.
Oh,
It's kind of pretty flattens out up at the top. It's not a sharp top.
yeah, I'm looking for micro ones that are.
Yep.
Don't take much, a couple meters.
Come down, you said?
Nope.
No.
Just zigging and zagging as we come to the top here.
So is this current
About at the
pushing
end of your
down
leash.
slope or all the way, or just across?
What's that?
Is the current coming down the slope or across the slope more?
It's been coming from the south, but flowing to the north. But now it seems to be the other way. When we were just looking at that paragorgia, it was weather veining arc around to look west. So I guess that would be to the east. I know we're right at the kind of at the top here, so I don't know what's going on.
That's swirling around.
Swirling around, yeah. But it was flowing to the north while we were coming up there. big time.
Mm-hmm.
Or to the north and east, maybe. Seems like when we changed head to the north, it was blowing me east. But when we did our little dog leg down below, it was, we definitely had our nose into it.
Yeah.
Looking west, and it was pushing me north, so. I don't know if it's tidal at this depth, or you think it's constant. So.
Not sure. It might. There will certainly be a tidal component even at this depth, but how much of that is also just constant or part of a bigger, like wind-driven current regime is, I have no idea.
Step to the west, 10 meters there.
Roger.
Chase these guys around.
Bridge nav. Let's go 10 meters west. Thank you.
Passed over another little mini Christmas tree coral, so we're definitely getting up into that habitat zone a bit more.
So are you meant to, do we want to get to the top of this feature, or do we want to skirt around on the slope?
Good question. Let me update the point so I can see where we are relative to some points.
How far are we from the top?
About 100 meters.
Right.
I think, yeah, we can, I think we should go to the top. That's going to be where the predictions for the highest diversity of corals are.
Go to the top and then kind of follow it along track and towards 883. And then if we feel like popping down the slope either way, we can.
Yeah, we can do that too. If there's any really cool habitat